today we're going to talk, my part of this is going to be regarding multiple sequence alignment and phylogeny inference, you guessed. What am I talking about when I discuss that? Oh, cool. That work? Okay, so the last time I talked to you, we talked about this. One of the things I talked about was performing sequence alignment. You get these alignments of a couple of sequences, which I discussed requiring some amount of work to actually perform the sequence alignment, but don't worry about the work, just worry about the fact that you can produce an alignment of two sequences that in some way is aiming to recognize which parts of one sequence are related to which parts of the other sequence. That's largely what the outcome of a sequence alignment is. And then I pointed out to me the more complicated challenge, which is not just to align one sequence to another sequence, but to say, I've got this one sequence that's in my hand, right? And I'd like to find other sequences that are related to it. Maybe I don't know what it is at all. This is how I presented it last time. And I want to find out what it is. And so I'd search a database to see if I can find anybody else who's similar to it. But another way I might approach this is I might have one sequence in hand and want to find a bunch of other related sequences because, as you'll see during the course of this talk, maybe having a bunch of other sequences is also helpful to me in some way. So you can imagine in the case I'm going to show in just a moment that you're interested in heme-containing proteins. These are they're responsible for binding or transport of oxygen. Okay, and you've got one globin protein in hand. That's a heme-containing oxygen-binding protein, and you use it to search some huge database, not just 8 sequences, but 500,000 sequences or whatever. You get a bunch of sequences back. You've searched this database, and most of them don't match because they're not globins, but a few of them do, and this ratio is ridiculous. It wouldn't be 3 out of 8. It would be 1 in a few million, but that would be a lot of small boxes with very few green check marks. So you have some fraction of them come through, and then you end up with a bunch of sequences, and maybe there's a horse myoglobin and a yak hemoglobin A and a rabbit hemoglobin B and a bunch of other matches as well. There's a few that you might have gotten. I've just got 45 that are gathered together in a sequence file, and you can see that I've got a bunch of them. And if you look carefully, you'll see that they're not all the same length. Some are shorter, some are longer, although the last bunch that are there are all very similar to each other, so they're all about the same length. And you might say, okay, great, I've got those. What do I do now? Cool, I got a bunch of homologs to my proteins, and I've got some, not just all hemoglobin A's. I've got hemoglobin A's and hemoglobin B's and myoglobins, and a few other globins fall into that mix as well. And the title slide was the hint of what you might do next, which is you might perform a multiple sequence alignment. And here's where I get to go off of the slides and actually show you stuff. So hopefully I can do this with the number of hands that I have and with the screen sharing technology that I've got. See how this works. Was it? It happened. Excellent. Okay, so I've got globins. This was that globin sequence file, which it looks like it's going there. Great. Lots and lots of globin sequences. They're not too long. And you could probably, if you were willing to spend a very long time, try to figure out, like, okay, how do these two relate to each other? Well, great, how do those two relate to each other? And you could, on a piece of paper, kind of work out what how they're all related to each other in some large collection, and you would waste years, or at least months of your life doing that. Or you could open up a terminal and clear this out so it's a little clearer. Is that font big enough to read? Do I need to make it bigger? Should it be bigger? Anybody need it to be bigger? Raise your hand. Cool. Okay. So then you can run a program like MAFT. MAFT is my go-to for performing multiple sequence alignment in almost all cases. Okay. And you can hand it the file, the FASTA file. That's what this format is, where I've got a greater than sign and then some name and then one or more lines afterwards that have sequence before the next greater than sign, which is another an indicator that's another name field. That's a FASTA formatted file. So I can hand it this file that's got 45 globins in it. File name is just my way of knowing I've got 45 globins in it. It's not a requirement that you have the number in the file name. And when I run math, I can hit enter, and it worked, and it's really fast for 45 short sequences like this. And sadly, it dumped all the sequences out to standard out, so it's right there in the terminal. That's not all that useful to me, but that's cool because I can just redirect the output to another file. And this is a fairly common naming convention, is to say, hey, this is an aligned FASTA file, an AFA. Not a, again, it's not a law that you have to name it that way, but that's a fairly common way to name things. Then when I do it, I can look at that file. There's a great example of where it would be better to have two hands, but I don't, or three hands, I guess, but I don't. I do have two. And here's what that alignment looks like. Notice it's still a FASTA file, formatted file. It's got a name and characters and name and characters, but I now have some dash characters, right? And those are somehow an indication that this sequence doesn't have characters there, even though some other sequence does, right? So that's a deletion relative to the characters that are present in other sequences, or maybe it just means that one other sequence had an insertion relative to these others. And boy, that's pretty hard to make sense of, right? I mean, you can kind of get it. With this one, it's not too bad because there's not a lot of insertions and deletions. But there's a lot to kind of make sense of here. And so the next thing you're likely to want to have, can everybody hear me? Okay. The next thing you're likely to want to have is some way of looking at these multiple sequence alignments that's a little bit more helpful than just a text file editor. I have a request. If you're going to have a conversation in the room, can you please do it in another room? Or maybe. If you're going to have a conversation while I'm lecturing, can you please do it in another room? Okay. Okay. And so what do we do? Well, there are several ways of viewing multiple sequence alignments, but one of my favorite is a piece of software called AliView. Whoops, I don't even have to do that. I can just tell it to open it. That same file. And AliView is this program that's going to come up on some screen here, on this one, but I can make it go there. It's this nice graphical visualizer of multiple sequence alignments. AliView can be installed on Windows or OS X or Linux. And you can see here, I don't know why it popped up so small, but you can see here, right, these are the sequence names that corresponded to the sequence names that were present here with the greater than sign lines. And then I've got all of my sequences afterwards. And you can see positions, the very beginning of the alignment to the very end of the alignment, and you can see that there are a few of these sequences that have whatever that is, 5 or 6 columns, 5 or 6 positions, amino acids that the other sequences don't, and that's also true for this batch of sequences here. If you were to look pretty closely at it, you would see, right, that all of these, I'm not sure if you're able to see the names here, but these are all of the hemoglobin A's. Right, all have something missing here, whereas the myoglobins all had something present there, right? And so maybe this isn't surprising that the hemoglobins are all more similar to each other. Hemoglobin A's are more similar to each other than the hemoglobin B's, and the myoglobins are all more similar to each other. But it's sort of right, this is a fairly easy family to look at, but it's sort of a clear reminder that, gosh, myoglobins, hemoglobins A and B, probably all originated at some common ancestor to this whole collection of, I believe these are all mammals. Yeah, Daniel. So I'm not sure if everybody could hear that. So the question is, does MAFT provide this beautiful ordering? Because it's really convenient that these guys are all clustered together and these guys are all clustered together. MAFT can either emit the set of sequences in the same order as you put it in, or it can emit them in the order that it chose to merge sequences together. And we're going to talk about that at the very end. And that order is going to tend to look like this. So it's going to tend to group similar sequences together. And I believe my input order is already ordered in this way. So I made it easier for MAFT to get it right. And I did not call it with the flag that causes it to reorder them according to the tree. But it can be made to do so and generally work pretty well. In this view, you can see some things that I think are maybe going to be fairly clear. So here's a column right near the end that's got a whole bunch of, won't zoom in for you, but R, right, arginine. So for the most part, that seems to be a highly conserved amino acid. Some of these others are very highly conserved. This is a column that's almost entirely all F's and B's, but sometimes I's. So you like to have a valine, but you're okay with an isoleucine and so forth. And some of the columns are much more conserved, and some of them are just sort of conserved within one group. All the hemoglobin B's have an H there, but not the other globins. And you can see very clearly some patterns of conservation where some columns are really not at all conserved, which suggests that maybe the ones that are conserved have something to do with the function, and maybe the ones that aren't conserved doesn't have much to do with the function. This shouldn't be a surprise, right? And this is a reasonably good way to be able to get a sense of what your active sites are, or something that's really vital to forming the structure, right? Like if all I need to have is an alpha helix in a particular region, then it doesn't matter which amino acids I use to get it, as long as I can keep the alpha helix. But if I need to have a particular atom out at a particular spot in order to be able to create some kind of an interaction, then I really can't mess around with the amino acid that's in that place. And so you can sort of see that with these alignments, but that's not the only way to be able to look at these, the information that you get from these alignments. There are a lot of different ways. I'm going to show you a couple just to give a little bit of a perspective of the use of multiple sequence alignments. And one way to do it is to make use of a tool called Skyline. This is cute because it's got an alignment and it draws things that look a little bit like towers in a city, like a skyline. Like great naming events of all time. And I can take that alignment file, and there are a variety of choices here. I'm just going to choose a couple and don't worry about the specifics of those choices. And I can ask it to generate the logo for that family, right? And I can see when I do that, that I get something that's where each position or column in this thing that's called a logo, right, is taller or shorter. And what that corresponds to is a measure of the deviation of a particular column from random chance, which is the specific name of that measure is relative entropy. But the important part of it is that here's this H, and if you went back and looked at the multiple sequence alignment, you'd see a column that's essentially all H's. Whereas here, I have both a multitude of letters, but also the column itself is shorter because there's much less insistence on a single letter being present in the column. So the more explicitly conserved a column is, both the taller the column will be and also the fewer letters there will be, and which letter is most common or most prevalent will be the letter of choice in a particular position. And you can use this to kind of get a sense of where are the regions that are more or less conserved. This is a pretty small alignment, but it can give you, in some cases, some pretty clear insight into where active sites might be in a protein. Okay. Another thing you can do is to use some method of identifying or placing the measure of conservation, which can also, there are a variety of ways of measuring conservation. Relative entropy is one, and there are others, and conserv uses a different one. I won't go into, but if you provide it with the right collection of, oh, thank you. Right now I have to be here anyway. If you provide it with the right collection of protein structure, where perhaps you know the PDB ID for one of the sequences that's in your alignment, or in my case, I happen to, I've already downloaded a structure from the Uniprot website for one of those, for the myoglobin for mouse, and I want to click on that and say, well, yes, I do have a multiple sequence alignment that is associated with it. If I don't, it will do a search against using the Hammer server and grab a bunch of sequences back and build a multiple sequence alignment using MAFT. So I've just shortcut all of those processes. And I'll say I do have a multiple sequence alignment, and nope, not that one. It's going to dislike that one. I want the AFA file. And then it wants me to pick which sequence I'm using, and I was using myoglobin mouse, if I remember correctly. And that's a wonky interface, but it works. Do I have a tree? I don't have a tree. Sure. I'm going to name this myoglobin mouse. And I'm not going to bother having it email me. But if you've got a thing that's going to take a long time, or if the server is being slow, you can have it email you. But in testing it this morning, it was running just fine. Takes a minute to run. And actually, even better than me having pressed that button and waiting for a minute, look, here it is. Nope, sorry. Here it is, pre-baked out of the oven. So it takes a minute or so for it to run for a reasonable sized input. And here's what the results look like. And it gives me a variety of viewpoints into something I can see about the conservation of the particular amino acids on this protein family based on the multiple sequence alignment. And here is one of those. This was the first few conserf results. Right? Why is it called conserf? Well, because it in some way gives us insight into, whoa, come on back. That was unfortunate. Into the conservation on the surface of the protein. And what I can see if I move its viewer around, we need to know what the legend tells us. Purplish colors are highly conserved, not purple is not conserved. So I get a pretty good sense that there are parts of this protein that maybe aren't particularly important in detail. They just sort of roughly have to stick in the way they are in order for the protein to work. And then there are parts that are probably a lot more important. There's probably a binding pocket sitting right there because it's all conserved. Maybe it's right here, right? And those purpley bits are the parts that seem to matter. And I would actually be kind of dissatisfied with just this conservation structure if I were really trying to understand the thing. I think I don't have enough sequences and enough diversity of the family to boil this down. There's a lot of conserved positions in this particular input. But if I get more remote homologs, I'll find that a much smaller number of the amino acids are going to be highlighted in purple, and I'll have probably a better sort of a tight view into what's actually conserved about it. But that gives you some sense of some stuff you can do with that multiple sequence alignment, right? You can build a multiple sequence alignment, MAFT. You can look at it yourself with AliView. You can use Skyline to get some notion of a logo, which can give you position-specific preferences and levels of conservation for those, and you can stare at conservation patterns across protein structures as well. Don't have to do it with myoglobin. I'm just going to take a quick look at another input because maybe you're interested in opsins, right? These are proteins that bind to light reactive chemicals of some sort and are the basis of vision and probably circadian rhythms. Let's see where she is. Circadian rhythms. I imagine you probably play with opsins at some point in your life, Cassandra. So I've got a pile of opsins that were the result of another kind of a search that was performed, and this is maybe it's a little bit bigger pile of them. And it turns out that if I run MAFT on this opsins file and I get an opsins AFA, it's still super fast. and I can open that file and I can view it in, nope, I already have it opt over in some random location. There we go. I can view it here. And what I'm going to see is something that's maybe a little less satisfying. I'm just showing this to you just because it's important to know that not all multiple sequence alignments look as beautiful as that Globin's input did. And there's a part of this that's really satisfying, right? Like these columns are quite nice. Again, you see some of these really highly conserved positions, these D and R. And there's several other sorts of positions that are, here's a column of mostly N's. And there are a few of these that are more highly conserved. And then you see regions that are scattershot all over the place. And if you were to work your way through this collection of like, what kind of opsins do I have? You're going to see that I've got some serotonin receptors, channel rhodopsins, and there's a much wider variety of these sequences where different families have picked up fairly substantially different domains or at least chunks of sequence that do whatever it is that they do, so that there's nothing in these sequences that looks anything like the things that are in these sequences, and MAFT ends up kind of some ways slapping characters together in ways that are probably not reliable. We'll talk about that reliability thing in a minute, but I can still take a look at the parts of this that appear to be conserved in some way. And there definitely are some parts of those by taking a look on the Skyline server. I could upload it again, and here's a pre-baked outcome of looking that up again. And you'll see that there's a much greater variance in the level of conservation at some positions that are really conserved, right, over a much larger diversity of sequences, and a whole bunch of positions that aren't particularly conserved at all. And so the columns are very tiny because, yeah, there are a few letters that happen a little more than chance, but none of them happens a lot more than chance. And this is going to give me, if I'm looking through it, it's going to give me probably a much better sense of what's important about this particular protein. There's a few positions that are probably associated with active sites, and the rest of it is not nearly as vital that it remain in a particular format. And I might be able to use that if I'm doing something with some of these options, and I really care about either identifying mutations that could be significantly problematic to the function of the protein, or being able to tinker with the protein in some way, tinker with its binding affinities for particular things. I have a little bit more insight into what's going to matter and what isn't going to matter. Conserv. Oh, that's unfortunate. I had conserv tabs. I guess I'm going to skip conserv on this one. Conserv there are other views into the conserv data that I had pre-baked, and apparently I closed the tabs on that look a little bit like looking at the multiple sequence alignment in alley view, but with coloring in the multiple sequence alignment that corresponds to conservation patterns. And what you would see is just something that mirrors this, is that there are a few that are highly conserved and most aren't highly conserved. Okay, what else might you do with a multiple sequence alignment? I bet a lot of us at some level are going to care about the history of a family, of a gene family, for example, right? So you might want to produce a phylogeny of that gene, a gene tree, okay? I guess this is a place where I can kill all of these tabs because I don't need them anymore. And I don't even need that anymore. And there's software that'll do that for you, that'll discover a tree. And we're going to talk a little bit more about trees in a bit, but here's a tool that is my quick and dirty preference, is FastTree. FastTree is a method, for those in the know, it performs maximum likelihood tree inference, but with a lot of heuristics to try to make it much, much faster, thus the name. So it's a pretty good place to start unless you need absolute clarity, in which case you can go to some slower programs. And with FastTree, I can hand it that same alignment file, options, and I can tell it I'd like it to spit the tree out to a file that I'm going to name .tree. That's, again, a fairly common suffix to add on to tree file names. I'm not sure why we use the word tree to store trees, but maybe we can all figure it out. So that makes a tree file, and cool, so I've got a tree file. Neat. Okay, that's what the tree looks like. Everybody cool? You can all see how all of the things are related to each other? Nobody can see what the relationships are between these, right? So that's why you want to have another program that will let you look at this Newick file format, and I'll describe what goes into this format in a few minutes, in a way that can actually help a human as opposed to a computer. I happen to have a program installed on my computer called Figtree, which I like to use. And what it does is opens in another window again. Knows where it has opened. No idea. Really weird. Figtree is there. Going to quick Figtree. Start it over again. Sorry. Okay. Now at least I have a window somewhere that I can drag around. So here is Figtree. It gives me a ton of options for laying things out. I can draw circular trees. I can draw whatever that is, or I can draw trees in a cladogram kind of a style. That's good. I can, alter the tip labels to make the font big enough to hopefully be able to read. Are you able to read those labels? Sort of. Okay, I'll say things about them in a minute, but oops, I didn't mean to do that. But what this gives me is a tree, right? I might drop that down a bit first. This gives me a tree which shows me that that there's a bunch of opsins that are all kind of out here, and then there's this human serotonin receptor and a rat serotonin receptor and another serotonin, a bunch of serotonin receptors are all clustered together. Maybe that's not a surprise. They're probably related to each other because they're serotonin receptors. And GCP and chicken visual pigment and stuff that's happening in the retina, and so the opsins that are in the retina, these guys are all clustered together in a tree also. But whereas some of these other opsins, if you look closely at what the species are often stuff that these guys are Drosophila, right? These are we've got insect opsins of some sort. I'm not sure we necessarily have the tree rooted in the correct place. Like, I'm not sure that these guys over here evolved from the same common ancestor as these, and FastTree doesn't really work that out unless you give it an outgroup, somebody who's related, but you know is the most ancient common ancestor. So with some care, I might find that I need to do something like reroot the tree, and probably that's where it is, because here I've got I've got a collection of animals that are not. I've got the insects up here and the other guys down here, and that's most likely where the split occurred. And you can see that you can kind of manipulate and work with the tree to gain some kind of inferences about the family of sequences that you're working with and see how things are related to each other. I'm not going to deeply belabor the point about how you can analyze this. I just want to highlight that there is a thing that you can do, which is you've got a multiple sequence alignment and you can produce a gene tree, a phylogeny that captures, it thinks, the evolutionary history, the history of divergence patterns of the ancestors of the extant sequences that you've provided into the multiple sequence and the phylogeny inference. Okay, that's what I wanted to show in terms of examples. Maybe it's worth asking now. Do you have questions before I continue to blather on about multiple sequence alignment? If you decide you had questions that you forgot to ask, feel free to grab me later. Yes? Consurf is free to use. Skyline is free to use. I made it. Everything I release is open source. Every tool that I've just shown you is a free to use tool. You will never see me demo tools that one has to pay for because I'm not a fan of that particular mode of doing science. I think that answered your question, yeah? Okay. Now, if only I could get, you may. Let me try to answer that, because I will definitely forget the first question if you ask a second question. So the first question, I think the big picture is it relates to, can I trust MAFT as being the truth? If I use some other piece of software, it may produce a different alignment. And how do I know which one is right? That's the key question. The answer is you don't know it's right. There's a good deal of benchmarking that's been done to try to understand the typical accuracy of various multiple sequence aligners. I've done a lot of that benchmarking. Others have done that as well. Typically, MAFT is the gold standard among these in terms of, on average, producing the most accurate alignments against known, compared to known alignments. But all multiple sequence aligners have errors. And there's a little cottage industry of some of us who have tried to develop methods for recognizing either whole alignments if one tool has done a better job than another tool. And I can point you to a couple of techniques that folks in the lab that I left behind have developed for trying to infer, given a collection of either which parameters were used or which tools were used, which of the alignments is better. There's also within that, it may be like you saw an example where where a lot of the columns were really clearly nice and probably right, but then when you start seeing insertions and deletions, things become a little bit more fuzzy, and it's not clear that it really got it right on the edges or in a part in the middle. And there's another kind of a framework of tools that aim to identify columns within a multiple sequence alignment that may not be correct, that have lower confidence and accuracy. And it's pretty common in downstream applications to take into account that measure of confidence. No, it's not pretty common. It's occasionally common. It happens sometimes. Not enough people actually do it. But some of us aim to take advantage of that and use that confidence or lack thereof to inform the downstream analyses that we do. But there certainly are, like the most straightforward thing to do is to say, I don't trust that column, throw it away. And that's a pretty common thing to do in phylogeny inference, for example. Does that get at your question, at the first question? Okay, and then the second question. Manually getting the alignment? Oh, edit. Sure. So if you have one particular gene that you're interested in, you perform one multiple sequence alignment on that, it's going to give you an alignment. And if you're an expert in that protein, you should always look at it and say, that is nonsense. That's not the way that particular 3 amino acids would be associated. It's clear that they're forming they're performing a helix turn or something. And so I'm going to shove them over to where all the other proteins are performing that helix turn. And so manual editing of a multiple sequence alignment is a completely legitimate thing to do, but it's labor intensive and knowledge intensive. And so if you aren't an expert in that protein, it can be hard to rely on that. And that's where, or if you're doing, and this is really important, if you're doing large scale work, for example, you're going to see some discussion about AlphaFold on Wednesday next week, I think. You're going to see some information about AlphaFold tomorrow, which is even better because AlphaFold is really cool. And that produces a multiple sequence alignment automatically and uses that automatically produced multiple sequence alignment to produce a predicted structure, you can interrupt that by manually editing it if you want to. But when they're doing hundreds of thousands of proteins across this collection of uniprot sequences, they couldn't afford to manually edit each one of those, and so you're just stuck doing the automated thing. But if you've got the time and inclination, you should always feel free to manually edit one. And the tool I just showed you, Alex, will allow you to pick and move and all alignments. That's one of the things they're designed to do. Did that answer that question? Great. Any other questions? Okay. So I just have a couple of slides about this. We've just predicted the first bullet point, which is that there are a variety of other considerations. I've got a couple of bullet points here. One of them is you might want to identify and trim problem regions, and this is the name of a couple of tools, and there's a small cottage industry of tools designed to do that. One thing that's quite common to do is that you might have a multiple sequence alignment that you've built previously, and then you find several new sequences, or you sequence a new organism, then you pull its sequences out, whatever it is, you've got some new sequences and you want to add them into multiple sequence alignment. There's not really a reason to rebuild an entire multiple sequence alignment, especially if you just manually edited it 5 years ago and spent 3 months of your life doing that. You want to use that and keep it intact. And there are methods, there are methods in almost all multiple sequence alignment tools to be able to take one existing alignment and just append other sequences onto that existing alignment. And in some ways, that's actually at the heart of what Hammer does. I've got HMM align listed here, is that it takes a collection that are aligned and then takes each new sequence and just aligns it to that. There are situations in which the entire, perhaps the entire sequences shouldn't be aligned. That's a poorly worded bullet, so let me give you a couple of specific examples of that. One of them is if the collection of sequences that you've been given, if they're all fragments of what the full length element looks like, and this doesn't normally happen if you're dealing with protein sequences, but if you're dealing with transposable elements, for example, where they either may have been placed into the genome in just a small fragment, for example, only if the retrotransposition starts at the 3' end and works back until the machinery falls off of the DNA, then you end up with something like this, where the majority of the instances are on the 3' end, and then fewer and fewer and fewer of them make their way all the way to the 5' end. And also, if it happened a long time ago and it's not a functional gene, it's not uncommon for deletions or recombinations to cause chunks of the sequence to go away. And so all of the instances that are in your genome are likely to be fragments of the full length element that once upon a time existed 50 million years ago or 150 million years ago for this L2. Okay, and that's a real problem. If you try to just do a multiple sequence alignment, you hand these sequences to MAFT, it may very well see some of these small sequences over here and fail to recognize that they belong lined up on the right, and it might try to either take the individual characters and piecemeal spread them out across the alignment in ways that make it happier, or just shunt it off to another side of the sequence. And the fragments tend to really be a problem when performing sequence alignment, and we've got a recently released paper discussing a newer method of dealing with fragmented sequences without getting into details. The blue line is our tool, and being higher is better in terms of a measure of accuracy as fragments become more and more of a problem as you go to the right. So most of these tools fall off to having really atrocious sequence alignment quality when you give them small fragments, but with care that can be overcome. Another example of parts of sequences that shouldn't be aligned, is if you've got two proteoforms or isoforms, alternative splicing products. So an example of this would be if you've got, the first gene uses this exon A, exon B and exon D, and the second one uses exon A, exon C, and exon D, then you really don't want to align, right? If these two proteins are being aligned to each other, you want the blue part, which came from one exon and is totally unrelated to the red part, the other exon. You really don't want to align them to each other. And if you do, if the alignment tool isn't aware of what's going on, it will try to shove those sequences together in whatever way it can. So this is an example of this QQIV should be aligned to nothing but muscle or MAFT or clusteral omega. All try to align it to other characters, and the same is true for many of these with this PTEAVL, which is coming from a different exon, which again should not be aligned to anything, but is frequently aligned to other things in some cases. And a tool that is aware of the fact that exons exist and accounts for them might, and in fact does, perform better than that. This is a paper that we've got from a few years ago, and I just got an email this morning with a draft of a new manuscript for the next version of it. That was a fun thing to see in the morning. Okay, so last thing to say about multiple sequence alignment is, I'm not going to dive into details about it, but I think it's nice for biologists who are using these tools to have at least an inkling of what it is that they're doing internally. You've seen me talk about what sequence alignment is doing, which is in some way trying to identify a best path through that matrix of this two-dimensional matrix where this dimension corresponds to one sequence and this one corresponds to another sequence, and that's a quadratic size matrix. If it's length n sequences, then I have to do n squared amount of work to be able to find that path. Ideally, if I have 3 sequences, now I have a path in an n by n by n matrix, so that takes n cubed time. And if I have 100 sequences, which is roughly what I had in the opsons, then that would be an n by n by n. I don't know how to draw the 4th dimension, by n, 100-dimensional space that I have to find the best path in, which is 2 to the 100 number of calculations that need to be performed. And that's, computer scientists in the room will have heard people say in the past, that's as many atoms as there are in the known universe, right? So that's a lot of calculations to perform. You don't want to do that. And so there are methods that are performed by multiple sequence alignment tools that definitely don't identify the actual best scoring alignment. They just try their best to come up with something that's pretty close. And the strategy roughly boils down to these stages, with constructing a merge tree and merging alignments and polishing the alignments being really the core of it, which I've highlighted in red here. I'm going to talk briefly about what that means. It's easier to sit. So we're given a set of sequences. Here I've got 7 sequences, and apparently it just moves forward without me. And the sequences are, gosh, what is it doing? We build a tree that's going to relate those sequences in some way, okay? I'm not talking to you about how that tree is built. It's just there's some way of building that tree. And then sequences are merged into groups according to the order that's suggested by that tree. And you can see me doing it with these cute little colored bars. And when I say merge, what I mean is they're aligned using sequence alignment methods like we've talked about. And when 2 leaves are merged, right, that just means align 2 sequences. And in the general case of merging, we align 2 alignments. Okay, here's an alignment and here's an alignment, and those have to be aligned together. And that's mostly done the same way as aligning 2 sequences, finding some path in a matrix, where the key is that the alignment, one alignment and the other alignment, they're forced to remain intact with each other. All of the columns in one are aligned to the columns in the other, and I've got a problem with the animation here. Sorry. These should be gaps. This column and this column and this column should all be gaps here and here, showing the fact that either gaps are placed as columns entirely or the columns themselves are aligned to each other. Those columns are kept intact for computational efficiency reasons. If you don't do that, things get much slower. But sometimes mistakes can be made. So for example, here, there's a choice that T could have been aligned to either this G or this G, and it wouldn't change the score of the best scoring alignment of doing that. But it turns out that if that it wasn't necessarily the right choice. And we're going to see that when this alignment gets aligned to this other alignment, that it would have been better for the T to have been over here. Right? So that's a thing that this heuristic approach to performing the alignment of two alignments needs to recover from, and the strategy for dealing with that is to do something called polishing of the alignment, where the elements, the individual sequences in this alignment are going to be split in some way, and that way can be random, arbitrary sort of splitting. There are a variety of approaches to doing it, and then those are then realigned to each other into some new alignment that might have fixed such a thing like getting that T over to the correct place, and then you do that a bunch of times to try to recover. And that process that I just described, which skipped right through, is the general process that essentially all multiple sequence aligners go through with some perturbation on the basic idea. That's multiple sequence alignment. That's all I'm going to say about it, but I'm happy to talk more about multiple sequence alignment offline. Got more questions. The last thing I want to talk about is just kind of a quick discussion of this phylogeny inference thing. I showed you phylogeny inference in action, right? Run fast tree, get a Newick file format that you can't read yourself, but you can stick into a tree viewer like Fig Tree, and then you imagine that you can gain some kind of inference of what's going on with the phylogeny. Let's talk a little bit more about that. There are a variety of approaches that can be used, and they tend to be clumped into a few classes. The generally computationally simplest approach, approaches all fall into what's called a distance-based phylogeny inference. This is just a hierarchical clustering approach in which you've got a collection of things. Here I've got 7 sequences, and they're named A, B, C, D, E, F, and G. And some method has been used to compute some measure of distance between those sequences. So sequence B is 19 away from sequence A, whatever 19 is. That might be number of mutations, or it could be some other scoring metric. And then from that distance matrix, some repeated process of pairing things off will be performed. So you'll get a cluster of 2, and then another, maybe a cluster of 2, and then those 2 might be clustered. So then you have a cluster of 4, and this creates a hierarchy of clusters. And you can see that sort of happening with B and F being merged first. They're getting merged first because they have the smallest distance between them. And then maybe that will result in some modified distance matrix that accounts for the fact that B and F were joined. And then maybe A and D are merged next because they have the smallest distance between them. And the process continues forward like that. I'm not going to try to teach you how hierarchical clustering works with an actual worked example, but I'm happy to discuss it, and you can actually hunt it down, the details on Wikipedia or elsewhere. What I've just shown you here is actually a method called UPGMA. Neighbor joining works a lot like it, but it's got a slightly more complicated function, and it ends up sort of inferring internal points in some tree so that it takes the set of sequences from what amounts to a star tree where it says, I don't know how they're related to each other, and slowly accumulates them into an increasingly good bet for each of these of what their history was. And this is proven to be correct under particular conditions that you can describe for input sequences that almost never hold for real biological sequences, but they're provable about something. Another strategy, there's another related method called parsimony, which is, I think, no longer worth even talking about because it's been superseded by maximum likelihood approaches, which effectively do the following. They say, for every possible tree, and I'm not going to tell you how many there are, but I will tell you that the number of trees that are possible for a small number of sequences is off the charts, so you can't actually do this. But roughly speaking, it's for every possible tree, and worse, within a tree, for every possible branch length combination that you might have, then you've got a tree. And so I'm drawing a kind of a tree here, right? Then for each column in the alignment, say, okay, well, what's the likelihood related to the probability of these of this tree having been responsible for this particular collection of characters? Okay, and that boils down to to computing the probability of observing a substitution from some ancestral character to some new character along a branch of some particular length, given some evolutionary model. That all of those things are parameters that go into the calculation of this, and they're all things that either you give them or you can learn in the process of maximum likelihood, and it can become a quite computationally expensive exercise, partly because it's a little bit expensive to do that calculation on a given tree, but mostly because the number of trees is super exponential. So there are a lot of cool heuristics that are used to try to build an individual tree and then keep flipping branches around on that tree, repeatedly trying to improve the tree over and over again. This is what tools, and then the goal is to pick the tree that's got the highest overall likelihood of the ones that you've tested. This is what's used in the tool RaxML, which I would say is your go-to if you're looking for the highest quality maximum likelihood phylogeny inference tools. It's also what FastTree is largely aiming to pull off, although it's got some quick heuristics that tend to make it approximations of what RaxML is doing. But oh my God, it's so much faster. So it all depends on what you're trying to get at. That's not the only kind of phylogeny inference method out there. There's another really important generic class of strategy for recovering phylogenies, which involves a thing called Bayesian inference. Specifically, there are approaches that involve sampling via a sampling approach called Markov chain Monte Carlo, which essentially picks a random number or a bunch of random numbers, and using those random numbers decides how to perturb a tree in some way, and based on that perturbation computes a likelihood. It's the same thing as the maximum likelihood is doing, but rather than always trying to hill climb and find the best thing, right, it just accepts the new tree if it's better, and if it's not better, it still potentially accepts it with some probability that is a lower probability if the tree is much worse, and a reasonably small probability, but not tiny, if the tree is only a little bit worse. And what you end up with is the ability to say, if you think of this as a probability landscape that I'm showing on the slide here, if maybe I got unlucky and I started at this position right here, if I'm using a maximum likelihood approach, what that's going to do is it's going to hill climb. It's going to work its way towards the best that it can get, and it's going to end up at some tree that's got this likelihood. Right? This is the local maximum that it can find, and then it's going to get stuck. And so maximum likelihood suffers from this problem of initiation, whereas Bayesian inference approaches such as in Mr. Bayes or Beast will certainly sample that region, but they also have the potential to work their way back down the hill, wander around, and then find a larger hill or a taller hill at some other point. And the key to these tools is that you need to let them run for a long time. They're doing a sampling approach, and you need to allow them to run. This is maybe not so different from MD in some ways, is that you're doing a ton of iterations to be able to explore the space of what the probabilities are that support a particular choice. And what you'll end up with, you hope, is that distribution that largely centers around the peaks of the tallest mountains in this probability space, and maybe a little bit less so around lower mountains and not much at all down at the base of the probability space. Ballyphi is a particular case of Bayesian inference in that it concurrently computes both a phylogeny or explores phylogenies and multiple sequence alignments, and in some cases can produce some much more informative multiple sequence alignments, but it can only handle under 20 sequences, because more than that and the computational load of doing all of this exploration is just too much. Couple more slides. So I just wanted to put this in here. I showed you that horrible Newick file format that I asked if you could read, and of course nobody could actually read it in the room. That includes me. I kind of can, but I would have had to stare at it for a long time. The format is something called a Newick format, and you can see a tree here. This tree probably you can make sense of, right? A's nearest neighbor is B, B's nearest neighbor is E, C's nearest neighbor is A and B together, right? The subtree that contains A and B. And in a Newick format, it just uses the names, commas and parentheses to represent that same connection. So A comma B, well, that means A and B are neighbors, siblings. D comma E, they're siblings, and you put parentheses around both of those. And then you can see that this AB pair, comma C, oh, so A and B, that subtree, its nearest neighbor is this one C, parentheses around it, and that collection of 3, comma, this collection of 2 is connected. And so that's all Newick format is. And so it's just a nesting format that uses parentheses and commas to show the relationships as opposed to drawing a picture. It's a computer readable format that allows computers to be able to draw this format. It doesn't have the spaces. That's just so that you can see how the A and B and C and D line up. And I want to mention a couple of considerations about phylogeny inference. There are lots of them, but I'm going to mention a couple of them. One of them is you've got to think about visualization, and Figtree is one, and there are a variety of other visualization tools as well. Some online, some that you can download. You already know that I like Figtree, although it's pretty old at this point. Another really important consideration is this topic of the difference between a gene tree and a species tree. So what I've shown you in the examples I showed, like the opsins and the globins, those are gene trees, right? Those are, for this gene, what is the inferred evolutionary history of the particular set of sequences that I added in? But that didn't really tell me anything about the species. And in fact, if we went back and looked closely at them again, you'd see that there are globins for a couple of different humans and for mouse and for rat and for horse. I think I had all 3 of the myoglobin and hemoglobin A and B were all there. And I don't think any of us would like to think that horses evolved 3 different times, right? And that they had a common ancestor with mouse in one, and then also in another, and also in another. That would be weird to think that there are that many. So these are just, the gene trees aren't necessarily directly tied to species trees. And in fact, even if I'd had only one horse in each one of those, and one mouse and one human and so forth in that collection of sequences, that still wouldn't necessarily have been the species history, right? The species tree. And I've placed this picture here to highlight the notion of incomplete lineage sorting, which the picture is a little bit complicated, but I'll try to get at it for a second. The big, thick black lines, many of you will have been exposed to this, but not all. This corresponds to what we might think of as the speciation. Well, let's imagine that a river separated the individuals who were in this group here, such that the guys on one side ended up in group A and guys on their side ended up in group B. Then you wait a few million years, and now A and B are now speciated, diverged from each other sufficiently that you'd call them different species. But there may have been some polymorphism in the population. Relate to something I think Asif is talking about. Well, okay, no, I guess we talked about variants in the last time, right? The SNPs and such. So the polymorphism that may have been present in this population, such that the lineages weren't sorted. There wasn't a fixed selection of one versus the other within that population. And then it may have been later that the actual fixation of one gene within the population occurred, such that if you looked at the gene trees, right, you might see that some of them agree that A and B are nearest neighbors, and then C is the outgroup. But for another gene, it might be that B and C are nearest neighbors, and A is the outgroup. And looking at an individual gene tree doesn't necessarily tell you what the species history was. You need to do something to take into account the collection of gene histories. And that can be either by computing a bunch of trees and then using methods that merge these trees together, producing what's called a super tree, or by another approach that's often taken is to just concatenate all of the sequences for a whole bunch of genes and then hand that into a tool, and that's called a super matrix approach, and there are good reasons to use both. Last thing to mention, and then I'll shut up and let Asif start talking, is that why do you want to do phylogeny inference? Well, one of the reasons would of course be that it's useful on its own. You might be able to infer when some particular feature arose, or look at the the speciation patterns of a clade of hosts and their endosymbionts, for example, and see what if there were any kind of a transfer of symbionts. There are a million things you can do with the phylogeny itself. You might be able to use it to compute some measures of position-specific conservation, so DNDS scores or phylop. You can compute conservation at the nucleotide level. You can identify with the NDS regions that are undergoing positive selection and a variety of other sorts of things that are related to conservation by itself. And it can aid in, for example, structure prediction, either the prediction of protein structure, two-dimensional or three-dimensional, or RNA structure, same thing. And my slide here says Wednesday because that's when I thought it was going to be, but I guess it's actually tomorrow. You're going to learn about the utility of multiple sequence alignments towards predicting protein structures in AlphaFold. And that is where I stop. So I'm going to answer any questions if you got them, and then I can hand the microphone over to Osif. And I don't mind answering questions offline. What I'm here for. Yeah. Oh, I didn't get into those because yeah. So the question is, what do I think of bootstrap values? So I guess the reason I didn't get into them is that I was sleepy when I was making some of the slides, and I didn't think to add that in. That's a really important thing that I've left out, and I'm a little embarrassed about it. Performing phylogeny inference, right? I give you a sequence alignment and infer a tree, and I hope it's right. But just like with sequence alignments, I don't know that it's right. I just have sort of like a maximum likelihood approach. And the method that Jack is mentioning, which is bootstrapping, is kind of an ancient statistical inference approach in general, which aims to say, look, what I'd love to do is have had an infinite number of data points that could tell me what the relationship between this collection of elements were, right? The collection of genes, for example. But all I have is this alignment that's 400 characters wide. I want trillions of characters. That gives me the most statistical power, but I've got 400 characters wide or 1,000 or whatever it is. So what can I do? Well, I can't sample more amino acids from a longer protein because this is the protein I have. But what I can do is something called bootstrapping. Bootstrapping is like pull yourself up by your bootstraps. Somehow you're supposed to pull yourself up. So bootstrapping is an idea that says, well, okay, well, I've got 400 columns. What if I believe that those were sampled from this infinite distribution? So I'll just sample from those and hope that they accurately represent the distribution. And so if I had 400 columns, I will sample with replacement 400 columns for a new alignment, right? That means that column 1 might get used 5 times in this sampled one, and column 2 might not get used at all. And I'll get some new alignment, okay? And I'll infer a phylogeny with that. And I'll sample it again, right, from the original one. And that's going to give me a different collection of sequences or of columns where some get used more often and some get used less often. And I'll build another alignment or another phylogeny. I'll do it again and again and again. I'll do it 100 times maybe, right? And if I do it 100 times, then what I can ask is questions like, okay, if I've got a clade of these 4 species that all end up together, right, in one tree, well, do they end up together in most of the trees or not? If they end up together in 95% of the trees, then I would say I've got a 95% bootstrap confidence value, right? Like when I tried building this, it might be that some of the columns in my multiple sequence alignment were noisy, but some of the times they get left out, and that noise stops messing up my phylogeny inference, and so I get things different. But if I reliably over and over again keep seeing the same tree that comes out of it, then I have more confidence in it. Whereas if bootstrapping sometimes leaves a part of the tree intact and sometimes doesn't, then I'm less confident that it's a real thing. And your question was, what do I think about that? And I think it's a great idea because it gives me some measure of how reliable a thing is. Bootstrapping doesn't guarantee, if you get a high bootstrap value, it doesn't guarantee that it's actually right? But it gives you a pretty good reason to trust the inference that you've just got. Did that answer your question? Okay. Thank you for making me say something about bootstrapping. Okay, so the question is, what would be the minimum percent identity necessary to be able to produce a meaningful multiple sequence alignment or phylogeny? And my answer to that would be that if you can detect their relationship via sequence alignment from the first slide, right, via something like BLAST or hammer, then I'm of the opinion that you ought to be able to align them. So what does that mean? Protein sequences that are 50% identical are essentially identical from a statistical perspective, right? There are only 50% of the characters are the same, but it's so clear that they're related to each other, and multiple sequence alignment tools should be able to handle that without a problem. Sequences that are only 20% identical at the protein level are still relatively identifiable and from a homology search perspective, and I would expect to be able to build multiple sequence alignments and infer a phylogeny from them too. The only place that I would say you wouldn't be able to is if you can't align them in the first place, right? So if I can get alignments of them, if I can get results from BLAST or hammer, then I'm comfortable throwing them in and building a multiple sequence alignment and phylogeny downstream. As diversity increases, it gets harder. There's an increased risk of getting things wrong in the multiple sequence alignment, which can then mean that you get more things wrong in the phylogeny inference, but with numbers comes power. Oh, and DNA, the numbers are much higher, right? Call it 50% tends to be about the point, 50% identity tends to be about the point where things just really fall off the map in terms of being able to recover related sequences. If there are more questions, I'm happy to answer them offline.